characteristics of bacteria. And this is part of our infectious disease unit. So, bacteria are one of the 6 different types of infectious agents. This is the infectious agent we're really going to focus on moving forward. So that's why we're going to delve just a little bit deeper into the characteristics of bacteria. So, to start, bacteria are prokaryotic. Now, for some of you, that's a new word. If you've had biology, you are hopefully familiar with prokaryotic versus eukaryotic. But we'll get into, in just a few minutes, what separates prokaryotic from eukaryotic cells. Now, bacteria cells are similar to our cells in several ways, but there's also a lot of really important differences. Bacteria have many unique adaptations that allow them to live in many different environments. So some bacteria can live in really, really hot temperatures, while others can live in cool or colder temperatures. The majority of bacteria typically live in warm or even room temperature environments. It just really depends on the specific species and where they've adapted to live. Then bacteria have evolved into millions of different species, so there are so many different types. We'll talk about a couple, but there's many, 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 many species. So this is just a diagram to illustrate the relationship between the different kingdoms and their origin. So bacteria are actually a completely separate branch compared to the kingdoms that you're more familiar with, so animals and fungi and plants and protists. So those protozoa that were one of our infectious agents, they would actually fit within this protist kingdom. But here's bacteria, all the way on the left, and here's animals, which is where we fit, all the way on the right, but we're still going to see a lot of similarities between the two. So, first of all, bacteria come in a couple different shapes and sizes, and they also are in different configurations depending on the species. So, we can't actually see bacteria, like a single bacterium, without a microscope. They are microscopic, super tiny. Just think about, how small a cell is, and that's, how small a bacterium is. When we view them under the microscope, they do have, or they will appear in one of 3 distinct shapes. So we have bacilli or bacillus, which is rod-shaped. And those would look like little Tic Tacs or pills. Then we have cocci or coccus, and those are spherical or circular. And then we have sporilli or sporillum, which are spiral shaped. They're longer than the bacilli typically. So those are the 3 main shapes that we would see. But, like I mentioned, bacteria do come in different configurations, or arrangements. You could also say arrangements. So, sometimes we see individual cells. You could have an individual circle, so a coccus, a bacillus, a sporillum. Diplo refers to 2 or a pair of. So diplococci are 2 circular bacteria, but you could also have diplobacillus. Staphylococci, so staphylo refers to a cluster, so just a group, cluster is really the best way to describe it. There's no specific, arrangement within the cluster. They're just kind of piled together. So you can have staphylococci, you can have staphylobacilli, the arrangement is independent of the shape. Then we have strepto. So strepto refers to a chain. So streptococci is a chain of circular bacteria, whereas streptobacilli would be a chain of rod-shaped bacteria. And then we don't really get into these two arrangements. So arrangement, you have singular, you have diplo, staphylo, and strepto. And then shape, again we have rod-shaped, which are bacilli, or bacillus, depending on the plural or singular. Cocci or coccus, which are spherical, and spirilli or spirillum, which are your spiral-shaped. So, I mentioned earlier that bacteria are prokaryotes. So there's two main types of cells. You have prokaryotic cells and eukaryotic cells. Or you could even go as far to say prokaryotic organisms and eukaryotic organisms. So let's just start with similarities. So prokaryotic and eukaryotic cells all have cytoplasm, and that's the fluid or the jelly-like substance that kind of holds everything in place. Both types of cells have a plasma or cell membrane, and that's the barrier around the cell, composed of that phospholipid bilayer. It really dictates what molecules move in and out of the cell, how they move. It's not always the outermost layer. Sometimes we see a cell wall or other barriers, but there will be a cell membrane present on both types of cells. Both types of cells have ribosomes. That's where proteins are put together. So if you think back to our protein synthesis unit, we have mRNA that travels to the ribosome, and tRNA deposits those amino acids, and that's where the amino acid chain is put together, which will eventually become a protein. So we need that process to happen in both prokaryotes and eukaryotes, and so ribosomes are present in both. And then both types of cells also have DNA. So in order for a cell to be considered living, or in order for something to be considered a cell, it has to have DNA or some type of hereditary material. So that can be some version of RNA, or there are actually different configurations or arrangements of DNA. So here is the big difference between prokaryotes and eukaryotes. Prokaryotes do not have a nucleus. And it's a little bit broader than that. They don't have membrane-bound organelles. So prokaryotes don't have mitochondria or chloroplasts, or like I said, they don't have a nucleus. So any organelle that's bound, by membrane, prokaryotes do not have. Eukaryotes are much more complex cells, and we're going to find a lot more variation of organelles within the eukaryotic cells. DNA in the bacterial cell is also different. So bacteria do have DNA, but it's shaped differently, and it's located in a little bit different space within the cell. So bacterial DNA is contained in one big circular chromosome that's just kind of floating in the cytoplasm, whereas eukaryotes have several linear chromosomes. So if you remember looking at the karyotype several weeks ago of our little chromosomes, we have 23 pairs of these little strands, whereas in bacteria there's just one big circular chromosome. The chromosome is located in a region called the nucleoid, but that is just a region. It's not bound by membrane. It's just an area within the cytoplasm. And then bacteria also have these small rings of DNA that we call plasmids. Plasmids are really important in biotechnology for a couple reasons. When we get into medical interventions and biomedical innovations, we come back and we look at plasmids and really bacteria in a lot more depth, a lot more detail because of the importance of bacteria and plasmids in medicine and biotechnology. Now, bacteria also have two other features, a cell wall and a flagella, or flagellum, and then some bacteria also have a capsule outside the cell wall. So we'll take a look at those in just a second. Okay, so a flagellum, that's the singular, flagella is plural, that is a tail, essentially. So it's just this little whip-like structure that allows the bacteria to move. Most bacteria are motile, or they can move. Then here is our nucleoid region, and if we unwound this DNA here, it would just be one really big circle. It doesn't look like a circle right now, but it would just be one large circle. Then you have the cytoplasm, that's just the blue area where everything's floating around. Our ribosomes in here, they're all over. Here's the plasmid. Again, you can't really tell it's a circle, but if we kind of unwound it, it would be a smaller circle compared to this chromosomal DNA. And we'll see a couple of those throughout the cell. They're not in the nucleoid region, they're out in the cytoplasm. Then we have our plasma membrane, a cell wall, and this bacterium also has a capsule. Not all bacteria will have a capsule. It's just included on this diagram to show you what that would look like. And then another structure that wasn't really mentioned on the previous page are these pili, or the singular version is a pilus, I'm pretty sure. These are not super important to you right now. I'll go ahead and tell you what they do, but we don't really focus too much on these. The pili allow the bacteria to really communicate and even attach to other bacteria to exchange DNA or genetic material. That's something we focus more on in the upper level courses, but that's what these are. Sometimes they're confused for cilia, which are another means of movement. They're like little projections around the outside of a cell that also allow for movement. But pili are for communication between and even transport between bacterial cells. So here's just a couple examples of some different bacteria. You have Streptococcus pyogenes. So if you look at this name, Streptococcus, we have chains of circular bacteria. Now this specific strain of bacteria is what causes strep throat. So if you've ever had strep throat, this is the bacteria that was infecting your throat and tonsils. Then we have Staphylococcus aureus. Again, look at this Staphylococcus. So we have a cluster of circular bacteria. And this, I believe, is your staph infections. So if you've heard of staph, this is typically the type that is causing the infection. And then this one is an example of a bacterium that the name doesn't necessarily reflect the shape. So this is Treponema pallidum. And I can't off the top of my head tell you what type of infection this causes, but this one is an infectious bacterium. And then this is E. coli. Again, the name doesn't really tell us about the shape, but this is, if you've ever heard of E. coli, this is what E. coli looks like under a microscope.